Hello and welcome to Molecular Views. I'm Dr. Cross, and today we're going to be discussing psychedelic biochemistry, or at least an introduction. I made this video because psychedelics are becoming more and more popular, especially natural psychedelics from plants. And today I want to give you some insight into exactly how they work with a molecular view of psychedelic biochemistry. You can reach me by email or on the World Wide Web or by my social media links. And hopefully, you'll have tons of questions when this is over, and you will join me for a more in depth class through Patreon. Let's first discuss what are drugs. Well, drugs can be anything that affects you physiologically or psychologically. And this is the exception of food and water, of course. So, drugs can be plants, they can be pills, they can be inhalers, they could be just about anything that's going to change your body chemistry. Now, what is a psychedelic? Well, psychedelic drugs, the term psychedelic, is Greek for mind revealing, offering insight into the consciousness, it's theorized by psychologists and psychiatrists. This typically, they say, involves a consciousness expansion or a change in consciousness, but nobody can ever really define what that means specifically. The chemistry of this is psychedelic molecules often are activators of serotonin receptors, specifically serotonin 2A, but also many psychedelics affect serotonin 1A and 2C receptors as well, and bind with really high affinity. When these molecules bind, they elicit their effects. So when we say something's binding with high affinity, it's eliciting an even stronger effect. Now, the key thing to remember about psychedelics is that we know that these substances can be hallucinatory or not. It just depends on what psychedelic you've ingested. In fact, you may be taking a psychedelic drug right now and not even know it. And we'll discuss why that is later. There are many popular natural psychedelics, and a few of them are extremely popular and have been legalized throughout the United States. The first most popular one is cannabis, marijuana. Yeah, pretty much everyone's done this psychedelic. The second most popular are psilocybin mushrooms, which now have even been legalized, much like cannabis in Western states. The third category are the tryptamine containing plants. These plants typically contain DMT, which I'm sure many people have heard of by now through Joe Rogan podcasts or Vice, TV shows. Tryptamine containing plants exist all over the world and even can include your lawn grass. Yes, your lawn grass most likely has DMT to some amount in it. So, tryptamine containing plants are very common and thus making it in third place here. Beta carboline containing compounds contain reversible MAOIs, meaning monoamine oxidase inhibitors. And we'll talk about why those are psychedelics and actually talk about some of the clinical applications that are using MAOIs for different kinds of therapies. And then we have the phenylethylamines. These are your peyotes, your San Pedro cactuses, and other mescaline containing cacti. And then finally, one of the most exciting but least known about is iboga or ibogaine containing plants. And this is just one genus of plant. So this is a very special plant, and we'll talk about it at as much length as we can at the end. So here are the basics. Natural psychedelics come from all over the world and have their own rich history. Every single one is cultivated differently, has a preferred climate, and a preferred geography. Psychedelics experiences require precautions and preparations to be taken because these experiences go through three different phases, which I talk about more in my psychedelic applications course. These three different phases include a rise, a peak, and a fall. During a peak experience, the user might find that they are unable to complete normal tasks. And that's why preparation and precautions are always most important. Today in modern medicine, we actually have several applications for psychedelic compounds. And it just not just doesn't stop in the clinic. We can actually go to the psychological and spiritual applications that are being either formed now in Western medicine or the spiritual applications that are being rediscovered, if you will, by Western medicine. So let's start off with everybody's favorite friend, nature's magical herb. Hi, it's the cannabis plant. Cannabis is actually not a psychedelic itself, but it contains the precursor for a psychedelic compound known as THC. THC is a psychedelic compound because it's the only cannabinoid that binds with those serotonin receptors we mentioned earlier. Every other compound doesn't seem to elicit an effect on serotonin receptors. Now, this plant is native to Asia, Africa, and we've even found a specific hemp strain to Australia. It's an angiosperm. It's in a family with roses, the Rosalis family, and it's a close cousin to hops, which is the plant used to brew beer. If we dip into looking closely at a cannabis plant, we'll note that there are all these snowy looking things all over it, and I think people are really drawn to the plant for that reason. This is the most commonly used psychedelic, and these trichomes, that's what these little snow things are actually little tiny THC factories, they call it. But they actually don't make THC, they make a precursor to THC. And when the plant is heated up by baking or smoking, you're going to transform all that THCA into THC. And so if you look closer, right at the edge of this leaf, you'll kind of make out that these look like almost needles or prongs, but I assure you they have a sort of mushroom shape to them, and this is because they're made of two different kinds of cells. So before we look closer into the trichome, we will note that THC mimics anandamide, which is a cannabinoid your body already makes. Anandamide regulates your mood, your eating habits, and your sleep patterns. So when we're making THC in the trichome, what happens here is this little cell, which looks like a neck, can be very long or very short, the stipe cell, is going to send phenol glucoside into the disc cell, which then is going to go into the vacuole, releasing glucose and change into olivitol. This olivitol will be excreted into a secretory cavity along with guanylyl pyrophosphate. These two combine to make cannabigerol, which is a precursor to all cannabinoids. This cannabigerol can convert without enzyme to cannabichromene, THC acid, or CBD acid. And the THC acid or CBD acids can then be transformed or decarboxylated by heat into THC or CBD. Now, CBD we won't talk about today. It will be in our Introduction to Cannabis Biochemistry class, and we'll get in depth into how the different compounds are made. But for the purposes of this class, we're going to say this decarboxylation is key in making THC because decarboxylation as a reaction in chemistry is key to making any psychedelic, and you'll see it repeated again. So let's look closer at this very important decarboxylation reaction. THCA, and the A is for acid, so you're wondering, acid? Well, not an acid like you would think. It's a carboxylic acid that's circled in red. And what happens is when this is heated up, this leaves as carbon dioxide, and it takes its electrons with it. Now, this intermediate is going to break this bond, and what do we get? THC. Now, this takes a lot of UV light, like 4 hours or more of direct sunlight, or over 200 to 300 degrees Fahrenheit, to actually make this conversion of at least 80% of the THCA. And that takes usually about 15 to 20 minutes if you're heating it. So it's important to note that THCA itself will not get you high. It actually is unable to because it doesn't have the ability to bind or affect the binding of cannabinoid receptors to serotonin receptors due to having that carboxylic acid group. More about that in Introduction to Cannabis Biochemistry, and I really hope you'll check that out. Moving on, when you ingest cannabis, or rather THC, you can have an effect at 10 milligrams of THC. 10 milligrams of THC is a low to moderate dose, while 20 milligrams of pure THC would be a higher moderate dose. Now, once you go over 50 milligrams of THC, depending on who you are and what your tolerance is, that's about where you start inducing psychedelic experiences. And these psychedelic experiences can last from 2 to 6 hours and involve some paranoia. So it's important to practice breathing exercises, yoga, Pilates, meditation, or whatever really calms you down. I will note that while we say the duration of the psychedelic experience will be 2 to 6 hours, the effects can last up to 3 days. As someone who's experienced about 800 milligrams of pure THC, I had effects lasting about 5 to 6 days, and psychedelic effects lasting for about 15 hours. Once THC enters your body, it actually is going to get hydroxylated and then carboxylated. We just decarboxylated, now we're recarboxylating, and we're going to stick on these nifty molecules to make this water soluble and actually expel the THC through the urine. Now, this is why we can tell if people have taken THC by a urine test. Um, so that's exactly how it metabolizes. And that's actually for THC going to take a long time as opposed to other psychedelics, which come out of the body within a day or so. The metabolism of THC is slow because it's so similar to our endogenous cannabinoids that it takes up to 30 and even I've heard 60 days to pass a drug test. And that's because this metabolism reaction goes pretty slowly. And the THC sort of comes out of your body a little bit at a time through your urine. Now, on to our next group. And I'm going to mention an animal and a mold here. And I know I'm talking about plants, but we're also talking about mushrooms, which are fungi. So just to be correct and specific, we're going to get into indole alkaloids. And these exist in all kinds of plants throughout the world and in different continents. They're very much worldwide. Um, we have harmal, which contains beta carboline. We have the ergolines in morning glories. We have these alkaloids in these two plants here, Alstonia and Rowolfia, I believe it's pronounced. And then we have monoterpene indole alkaloids in this beautiful, I can't say the name, roseus plant. But then we have also down here some non-plants. So we have Psilocybe cuneus, which contains psilocybin and psilocin. We'll talk about that chemistry and going into this section. And then we have ergot. And if you don't know what ergot is, it's a type of mold. And this is where they synthesized LSD from the ergolines. So just a note about a synthetic psychedelic. It was actually engineered from ergot. Ergot was a problem in the medieval times in Europe. It was contaminating the rice and driving people mad. They were having hallucinations and seeing things. And so it's also part of history. So it's good to learn about ergot. The other non-plant here is the California, the Colorado River toad, or Bufo alvarez. It has bufotenin, or we just call it bufo, which is a 5-methyloxy form of DMT. So what I want you to notice throughout this presentation as we move forward is we're going to be talking about a lot of things that look like this chemical structure on the left And this is the basis for all of these indole alkaloids. So if you notice, it's kind of like a little, I don't know, beetle shaped thing. And then it has like this NH, nitrogen, and a hydrogen right here at position 1. And so pay attention to that because it's going to repeat over and over. And what you'll notice is what we attach to this is super important. What is there versus not there is a difference between a psychedelic drug and an amino acid. So it's very interesting chemistry as we move forward, but also very similar. So it's going to seem like it repeats, but it's not actually repeating. So psilocybin mushrooms are one of the safest psychedelic plants. Well, it's not really a plant, it's a fungi, so excuse me there, to be specific. And it does belong to a different kingdom than plants. Plants have their own kingdom. Now, these psilocybin mushrooms are all kinds of shapes and sizes. They're of global abundance. There's numerous species, and the reason we call them the safest is because they're very non-toxic, even though they're very potent psychedelics. And new research shows that psilocybin itself could induce new neural networks, which becomes really useful in medicine and psychology for things like depression or spinal cord injury. I mean, the applications are endless, so it's very exciting, the field of psilocybin research right now. So how do we make psilocybin? How do these little guys make psilocybin? They're not plants. So they start off with something a little different. Um, and the plants seem to kind of mimic this chemistry. So I always start with the mushroom because it's a little more cleanly defined. So as I said, that decarboxylation reaction was going to be important, and now it is. So what we're going to start off with is L-tryptophan. You may have heard of tryptophan. It's in Turkey. It makes you sleepy. Now, L-tryptophan does not get you high. Nobody's gotten high off of turkey, right? So what do? We have to have a decarboxylation reaction to remove this big old arm off of this little beetle that I said you're going to see a lot of. Got to remove this arm. It's too long, it's bulky, it's in the way in order to bind to things, bind to serotonin receptors. So we're going to remove that through decarboxylation. So whole carboxylic acid group, nothing new, we just did this. All right, and now we're going to methylate this amine group and open this up because we want to branch it out. Why? Because when we methylate this, we get dimethyltryptamine. That's DMT. So the plants, you'll notice the reaction is going to be a little shorter. So dimethyltryptamine can be oxidized. Once it's oxidized at this back end here, this hydroxyl group is going to pop up. And guess what? That's psilocin. And in order to store psilocin, which isn't so stable, it's going to have to be phosphorylated, which happens in nature. And so we stick a phosphate group on this OH group here, and bang, psilocybin. And here we go, amine at the 1 position, and you see the little beetle there. Now we have these 2 arms here, psilocybin. So actually in your stomach, you're going to have a dephosphorylation and move this backwards in order to get psilocin, because psilocin is actually how you get high off of magic mushrooms. Now, the important parts are always the applications. And while this is just a basic course, and we get way more in depth in the next session, we are just going to vaguely talk about the precautions here. Now, it's important to note that psilocybin is 0.5% to 2% of the dried weight of your mushrooms, the dried weight of your mushrooms. And a moderate dose is actually 1 to 2.5 grams of dried mushrooms per average sized adult. Even though people will tell you it's 3.5 grams, that's to elicit a huge effect. And an average size adult doesn't usually need 3.5 grams. Plus, we don't know if we're getting 0.5% psilocybin or 2% psilocybin, so it's better to be cautious and prepare always your mushrooms according to weight. Now, I have a super great tea recipe that changes your psilocybin to psilocin as you brew this tea. And it's incredible. And that's covered in our next session when we get into the real particular stuff. But the duration, you're going to see whether you have tea or you eat the mushrooms, which is the most popular. I'm not sure why either, because it's not the most enjoyable. So you're going to have a 2 to 6 hour window for a low to moderate dose. And if you take a higher dose, 3 grams, 5 grams, 10 grams, you're going to slip into the 6 to 10 hour high dose levels. And these trips can actually go into 18 hours for something like a 10-gram dose. So the precautions you want to take is you want to notice that this is a moderate intensity psychedelic. It can make you feel like you're dying, but you probably won't die. However, if you do eat the mushrooms, they will make you nauseous, most likely. And so it's good to know this and have something around where if you do feel nauseous, you can throw up, like a bucket or something like that. An experienced guide, like someone who's done lots of mushrooms and is able to coach you through it, is recommended your first time. And I don't care if you're a scientist or even a mycologist, because all mycologists agree, do not pick wild mushrooms. Picking wild mushrooms is extremely dangerous even if you know what you're doing. There are so many poisonous mimics for not only magic mushrooms but edible mushrooms that it's extremely dangerous to just go out and pick wild mushrooms. So please don't do that. And remember, you will not likely die. You will only feel like you're dying if you have a bad experience. Some of the things that Experience Guide can help you with is pushing through those moments. So reach out to friends, hire a psychedelic counselor, do what you got to do, but take precautions and be careful. So how is our psilocybes metabolized in our body, right? So we have the psilocybin. I told you it's going to go into our body and be dephosphorylated. We're going to take off that PO4 group and we're going to put that OH back on there. Psilocin. Psilocin is actually what's going to interact with the serotonin receptor. And then once it's done there, it's going to come off. It's a small molecule, and it's going to go through the metabolism pathway by being made into what we call an acetaldehyde, and then into another. We're going to add another hydroxyl group over here, and then this, or the psilocin directly, is going to get that group added to it so that it's water soluble and can be excreted in the urine. So if you've used psilocybes within a day or so, it's going to be in your urine and you can be tested for it. So it's good to know. Psilocybin is actually being researched right now like crazy, and some of the medical uses are coming to fruition. It will now be about 60 years of psilocybin research in the United States. So this is really important step to cover. What we're finding is that low doses of psilocybin help anxiety and depression, while moderate doses are starting to help clinical depression and anxiety disorders and addictions and even cluster headaches and severe OCD. Now, the really high doses, like I talked about the 10-gram doses and things like that, they're still being explored because this looks like mostly spiritual endeavors, but also very important when you start to talk about mind spirit research. So we got to move on. I'm sorry to cut off the magic mushroom talk so quickly, but we have a whole host of tryptamine containing plants to talk about. And here I'm showing Psychotria viridis. This is also known as chacruna. It is a popular part of the admixture for ayahuasca. It contains up to 0.61% DMT in its dried leaves. And this is just a picture of DMT. As I said, I told you this little beetle looking thing would be an every molecular diagram, and here it is. And then N,N-dimethyltryptamine just has this with this methylated nitrogen right here. And chacruna is very, very simple. It's L-tryptophan decarboxylated to tryptamine which is methylated to dimethyltryptamine. Pretty cool. Now we're moving on to our beta carboline containing plants. This is where the whole monoamine oxidase inhibitors thing comes in. This is really cool because we do use monoamine oxidase inhibitors in medicine. They're being researched for tons and tons and tons of uses, but we don't really think of the ayahuasca vine as something that doesn't contain DMT, I guess. But it's true. The Banisteropsis caapi vine actually contains a reversible monoamine oxidase inhibitor. And this is important because without it, the DMT from the chacruna would just be chewed up in your stomach and you would have no great experience. There would be no psychedelic experience. You have to bring in Banisteropsis caapi or the Peganum harmala seed in order to get these monoamine oxidase inhibitors into the brew. And so what they do is they inhibit, again, the breakdown of DMT in the gut so that the DMT goes into your blood and you have that beautiful experience. So it's good to note. You'll also note that beta carboline looks kind of like a weird hybrid of the things we've been looking at. That's interesting too. There's a conserved chemistry here. But this is actually an enzyme inhibitor. So why are monoamine oxidase inhibitors really interesting to me? Well, they're interesting to a lot of spiritualists as well, because in our pineal gland, which is a very interesting part of the very center of your brain, we make two beta carboline molecules. We make pinealine and 6-MTHH, and these occur naturally in our brains. It's long been thought that these compounds play a role in our dream states and actually exposing us to some DMT during our sleep states so that we can dream. And if you notice, 6-MTHH looks a lot like harmine. They are not identical, but they are so similar it's hard to tell the difference. And harmine, again, is from the B caapi plant, the ayahuasca vine. And so it's really interesting. And as far as serotonin goes, I want to note the pineal gland regularly mediates our serotonin availability in our synapse. So basically, this is a secondary serotonin reaction as well. And the interaction of DMT specifically with the serotonin receptor is what we're going for. So using these in combination is really an ancient art. And there's so many recipes for these ayahuasca admixtures that it's just astounding. And it would be a lost art unless we actually got people really into this now. So it's very interesting to watch this modern history unfold because the mechanism of ayahuasca admixture is really beautiful chemistry. It's basically taking an monoamine oxidase inhibitor, ingesting it with the DMT to get the DMT to the bloodstream without having to inject it or smoke it. This makes it family friendly, and it's also a communal drug in communities that use ayahuasca. This is something you grow up taking and doing with your family and your entire community. This is a way of bonding. And the fact that people did not need giant laboratories to figure this out speaks a lot to the knowledge in nature. So now we can move on to my favorite category, which are the phenylethylamines. These are our mescaline containing cacti, whether they're peyote, San Pedro, Peruvian torch, Bolivian four-way torch cactus. These are really special to me because they're a little different from the other psychedelics. They do have a little bit of a different structure. They're based on a phenethylamine. A phenethylamine looks a little different. It's missing one of the ring structures, but it still has that little amine arm sticking out with the nitrogen and hydrogen on it, right? Mescaline itself actually has these little crab legs sticking out of it, And those are make it very similar to other hallucinogens, specifically the plant, like your DMT and tryptamines. Mescaline synthesis takes place in the cactus by converting several amino acids into dopamine and then converting that into mescaline through a series of O-methyl transferase reactions, and a couple hydroxylases, but you guys don't care about that. So when we make mescaline, again, we're starting with a cyclic amino acid base and we're changing the arms on it until it becomes something that's easy to store for the plant. Now, mescaline has been medically used for centuries, just like ayahuasca. Whole continents have used mescaline as medicine for as long as time has existed. In fact, if you look at Native American art, you can see the similarity in beading and patterns and colors throughout the American continent. Mescaline has been studied at length since, I believe, the early 1900s. Low doses have been noted to relieve anxiety and depression, much like magic mushrooms. Moderate doses, however, seem to relieve fatigue, which is different from other psychedelics. In fact, mushrooms contain more of like a mushrooms contain more of a sedating effect. So with mescaline, you're looking at something that's going to give you a little bit of energy at a moderate dose. And at a moderate dose, you can actually relieve some of the clinical level depression and anxiety. At high doses, we have lots of reports of actual physical healing and, of course, tons of thousands of years of spiritual endeavors. This is not research in big institutes, but also research on the ground with these communities. And so mescaline is really important because it was a medicine before medicine was a thing. And it also has a different kind of effect compared to other psychedelics. Um, People describe it as a mixture of LSD and MDMA, and I don't think it's exactly that, but it's a little bit different, more of a stimulant at a low to moderate dose. Like ayahuasca, ibogaine is a community drug that's often given to one person while the community surrounds them and practices healing ceremonies. Ibogaine is considered a strong psychedelic, and this strong psychedelic actually has been used to treat addiction, and not just any kind of addiction, opiate addiction, heroin addiction, because this particular psychedelic actually creates an environment in your brain where your brain starts to synthesize a brand new neurotransmitter. And that's really interesting. This novel neurotransmitter also stays in the body and is produced for up to 5 months, taking people through their most horrible times with addiction the first 5 months. Ibogaine is currently being studied by addiction experts and public health experts also. People have reported being relieved of all kinds of deep seated issues, including heart problems, from the use of ibogaine. And ibogaine is only found growing wild in Africa. So, this is another reason we must preserve our earth. The research does continue. As time moves on and progress is made, we ask new questions and rediscover old ways of healing. State by state, the United States is becoming cannabis friendly, cannabis legal, and even decriminalizing the plant altogether. In fact, some states and well, not provinces, but cities have began legalizing magic mushrooms in the United States. Plant psychedelics are increasingly becoming the subject of research programs in the United States, and synthetic psychedelics continue to be made. So let me give you some of my favorite books to read. If you're just starting out in psychedelics, it's important to get a humanistic view, but also know biology, chemistry, and tradition of the medicines you're using. PCAL and TCAL are a great introduction for Western readers because it tells the story of a man who discovers psychedelics from indigenous peoples and goes on a mission to create his own in a chemistry lab. Included is a beautiful romantic story, so it's great for all readers, and some recipes in the back of the book. Breaking Open the Head by Daniel Pinchbeck is a wonderful book to read if you're just one curious person about the psychedelic experience. The Sacred Mushroom of Visions is a great book about the roots of Tonanakato, and it gives a lot of, just a great resource on magic mushrooms and magic mushroom visions. And it's by Ralph Metzer. If you speak Spanish, I highly recommend Maria Sabina. It's a Spanish book by Enrique Gonzalez and Rubio Montoya, and it's excellent. Now, in our natural psychedelics workshop, if you keep up with us, we'll be going through cultivation, processing, and curation of these natural psychedelics. We're going to talk about organic plant and fungus care, harvesting techniques, safe handling and extraction, storage and use, using the medicine safely, including calculating your dosage, toxicity and precautions, and harm reduction. And if you give us a follow on our social media outlets, you'll get information on when those will come up. Additionally, if you join our Patreon, you're going to get that all included. Thank you so much for watching Molecular Views. This is Dr. Cross signing off. Bye.